điểm danh xong buổi này vắng hẳn nha.
Ôi, cái đông.
Thế này ở đâu ạ?
Ôi, cái quán này điểm danh very big.
Ngồi không điểm danh làm gì
cả ranh chu
vì
ra.
là học ở bậc đại học rồi đúng không? Mình tự xem là môn nào cần thiết với mình, cái gì cần thiết với mình tập trung sự thời gian sức lực cho nó, cái gì không cần thiết với mình thì thôi, đúng không? Đấy là mình lựa chọn các bạn, không ép làm gì cả. Thực ra thì mình cũng đánh giá cao những bạn mà biết cái gì quan trọng với mình, cái gì không quan trọng. Okay, quiet please, so we can come back to our talk. Quiet please. You can do whatever you want, but please do not make noise, alright? Quiet please. Please come in, find your seat quickly and make a stable environment of the class so we can start. So in Tuesday, we have gone back a little bit so in the history.
Tức là tuần này là sẽ còn điểm câu số 2 đúng không? Tuần này có điểm câu đúng không?
In the achievement of genetics, in 1953, when two scientists Watson and Crick developed find out the structure of DNA, the DNA molecule through the X-ray technology. In 2003, you guys was born in 2004. Okay, so in 2003, we have published the first draft of the human genome. I remember those days, every paper, every television program talk about it. Like it's one of the biggest achievements in the history of genetics, and indeed it is. But soon scientists realized that sequence the human genome is just a beginning part, which is an easy part, right? The most difficult part, the most complicated part is located in the behind, which is annotated genomes. Annotated genome is to find out what is the function of each gene, which part of gene encodes for protein, and what is the role and the function of this protein in the cycle of the organisms. Which part of the genome is controlled in regulation of which gene and which part of gene was turned off and which part of gene was turned off. And which part of gene is which actually junk, or what we call it, not have a significant effect on the life of the carrier. So up to date, today is 2023, we know pretty much about the human genomes. We know that the human genomes can identify physical appearance of a human. We know that some part of genomes related to the diet or the preference in your diet. For example, some people tend to consume more sugar than others. It's because when consuming sugar, normally in the brain, it will extract from, which is a substance, a signal peptide, bring to the consumer a satisfied feeling. Okay, and some people just have more receptor of this substrate more than the others. That means that when they consume sugar, the food containing sugar, it brings them feeling more comfortable, more satisfied feeling than the others. We know that the human genome also relates to the susceptible and resistant of certain diseases. For example, some people tend to have more risk with type 2 diabetes or lung cancer than the other. Some people are, in the other hand, quite resistant with lung cancer. They can smoke, they can drink, they can have an unhealthy lifestyle. But the risk with lung cancer is still lower than the others. And recently, scientists find out that the genes also affect on the sexual attractive person or model compared to the other. So for example, you hang out with your friend. You see some girl in the corners, you say, oh, look, guys, this girl is hot. But your friend said, oh, she's not my type. I'm feeling that she's not really attractive, okay? So science now find out that part of it, so let's say that love is always mystery in the human history, right? But now we have revealed that part of this reason can be explained by genes. And recently, a few groups have used also genome technology to identify a human face. So we just based on a genome to draw out based on what we know about the human genome to demonstrate how a person of this genome can be look like. So let's see the scientists talking about their research.
The next 16 minutes, I'm going to take you to a journey that is probably the biggest dream of humanity. Understand the code of life. So for me, everything started many, many years ago when I met the first 3D printer. The concept was fascinating. The 3D printer needs three elements: a bit of information, some raw material, some energy, and it can produce any object that was not there before. I was doing physics, I was coming back home, and I realized that I actually always knew a 3D printer, and everyone does. It was my mom. So my mom takes three elements. A bit of information is between my father and my mom, in this case. Raw elements and energy in the same media, that is food, and after several months, it produces me. And I was not existing before. So apart from the shock of my mom discovering that she was a 3D printer, I immediately got mesmerized by that piece, the first one, the information. What amount of information it takes to build and assemble a human? Is it much? Is it little? How many thumb drives you can fill? Well, I was studying physics at the beginning, and I took this approximation of a human as a gigantic Lego piece, okay? So you can imagine that the building blocks are little atoms, and there is a hydrogen here, a carbon here, a nitrogen here. So in the first approximation, if I can list the number of atoms that compose a human being, I can build it. Now, you can run some numbers, and that happens to be quite an astonishing number. So the number of atoms, the file that I will save in my thumb drive to assemble a little baby, will actually fill an entire Titanic of thumb drives, multiplied 2,000 times. This is the miracle of life. Every time you see from now on a pregnant lady, she's assembling the biggest amount of information that you will ever encounter. Forget big data, forget anything you heard of. This is the biggest amount of information that exists. But nature Fortunately, he's much smarter than a young physicist. And in 4 billion years, he managed to pack this information in a small crystal we call DNA. We met it for the first time in 1950 when Rosalind Franklin, an amazing scientist, a woman, took a picture of it. But it took us more than 40 years to finally poke inside a human cell, take out this crystal, unroll it, and read it for the first time. Well, the code comes out to be a fairly simple alphabet, 4 letters, A, T, C, and G. And to build a human, you need 3 billion of them. 3 billion. How many are 3 billion? Well, we don't really make any sense as a number, right? So I was thinking how I could explain myself better about how big and enormous is this code. But there is I'm going to have some help. And the best person that is going to help me to introduce you the code is actually the first man that sequenced, Dr. Craig Venter. So welcome on stage, Dr. Craig Venter. Not the man in his flesh. But for the first time in history, this is the genome of a specific human, printed page by page, letter by letter. 262,000 pages of information, 450 kilograms, shipped from the United States to Canada, only thanks to Bruno Bowling on stage, Lulu.com, a startup that did everything. It was an amazing feat. But this is the visual perception of what is the code of life. And now for the first time I can do something fun. I can actually poke inside it and read it. So let me take some interesting book like this. I have an annotation, it's a fairly big book. So just to let you see what is the code of life. Thousands and thousands and thousands and millions of letters that apparently make any sense. Let's get to a specific part. Let me read it to you. AAG, AAT, ATA. Well, to you sounds mute letters, but this sequence gives the color of the eyes to Craig. Show you another part of the book. It's actually a little more complicated. Chromosome 14, book 132. As you might expect. ATT, CTT, GATT. This human is lucky, because if you will miss just 2 letters in this position, 2 letters over 3 billion, you will become down to a terrible disease, cystic fibrosis. We have no cure for it, we don't know how to solve it, and it's just 2 letters of difference for what we are. A wonderful book, a mighty book. A mighty book that helped me to understand and show you also something quite remarkable. Every one of you, what makes me me and you, is just about 5 million of this. Half a book. For the rest, we are all absolutely identical. 500 pages is the miracle of life that you are. The rest, we all share it. So think about again when we think we are different, this is the amount that we share. So, now that I have your attention, the next question is, how do I read it? How do I make sense of it? Well, for how good you can be assembling Swedish furnitures, this instruction manual is nothing you can crack in your life. And so, in 2014, 2 famous texters, Peter Diamandis and Craig Venter himself, decided to assemble a new company. Human Longevity was born with one mission: trying everything we can try and learning everything we can learn from these books, with one target: making real the dream of personalized medicine, understanding what things should be done to have a better health and what are the secrets in this book. An amazing team, 40 data scientists, and many, many more people. A pleasure to work with. The concept is actually very simple. We're going to use a technology called machine learning. Okay? So on one side, we have genomes, thousands of them. On the other side, We collected the biggest database of human beings. Phenotypes, 3D scan, NMR, everything you can think of. Inside there, on these two opposite sides, there is the secret of the translation. And in the middle, we build a machine. We build a machine and we train a machine, well, not exactly one machine, many machines, to try to understand and translate the genome in a phenotype. What are those letters and what do they do? It's an approach that can be used for everything, but using it in genomics is particularly complicated. And little by little we grew and we wanted to build different challenges. We start from the beginning, from common traits. Common traits are comfortable because they're common. Everyone has them. So we started to ask our question, can we predict height? Can we read the books and predict your height? Oh, we actually can, with 5 centimeters of precision. BMI is fairly connected to your lifestyle, and we still can. We get in the ballpark, 8 kilograms of precision. Can we predict the eye color? Yeah, we can. 80% of accuracy. Can we predict the skin color? Yeah, we can. 80% of accuracy. Can we predict age? We can. Because apparently the code changes during your life. It gets shorter, you do species, it gets insertions, we read the signals, and we make a model. Now an interesting challenge. Can you predict a human face? It's a little complicated. Because a human face is scattered around millions of these letters, and a human face is not a very well-defined object. So we had to build the entire theory of it. We had to learn and teach a machine what is a face, an embedding, compress it, and if you are comfortable with machine learning, you understand what is the challenge. Now, after 15 years after we read the first sequence, this October, we started to see some signal. And it was a very emotional moment. So what you see here is a subject coming in our lab. This is a face for us. So we take the real face of a subject, we reduce the complexity, because not everything is in your face. Lots of features and defects and asymmetries are coming from your life. We symmetrize the face and we run our algorithm. Okay? The results that I show you right now, this is the prediction we have from the blood. Now, wait a second. In these seconds, your eyes are watching left and right, and your brain wants those pictures to be identical. So I ask you to do another exercise, to be honest. Please search for the differences, which are many. The biggest amount of signal comes from gender, then there is age, BMI, the ethnicity component of a human, and scaling up over that signal is much more complicated. But what you see here, even in the differences, let you understand that we are in the right ballpark, okay? That we are getting closer and is already giving you some emotions. This is another subject that comes in place, and this is a prediction. A little smaller face, we don't get the complete cranial structure, but it still is in the ballpark. This is a subject that comes in our lab, and this is the prediction. So, these people have never been seen in the training of the machine. These are the cool held out set, okay? But these are as well people that you will probably never believe. We're publishing everything in a scientific publication, you can read it. But since we are on stage, Chris challenged me, I probably expose myself and try to predict someone that you might recognize, okay? So, in this vial of blood, and believe me, you have no idea what we had to do to have blood now here. So, in this vial of blood, there is the amount of biological information that we need to do a full genome sequence. We just need this amount. We run this sequence, and I'm going to do it with you. And we start to layer up all the understanding we have. So in the vial of blood, we predict that he's a male, and the subject is a male. We predict that he is 1.76 m, the subject is 1.77 m. We predict that he's 76, the subject is 82. We predict his age, 38, the subject is 35. Predict his eye color, too dark. We predict his skin color, we're almost there. That's his face. Now, the reveal moment is that the subject is this person. And I did it intentionally. I I am a very particular and peculiar ethnicity. Southern European, Italians, they never fit in models. And it's particularly that ethnicity is a complex corner case for our model. But there is another point. So one of the things that we use a lot to recognize people will never be written in the genome. It's our free will. It's how I look. Not my haircut in this case, but my beard cut. So I'm going to show you, I'm going to just, in this case, transfer it, and this is nothing more than Photoshop, no modeling, the beard on the subject, and immediately we get much, much better in the film. So, why do we do this? We certainly don't do it for predicting the height or taking a beautiful picture out of your dog. We do it because the same technology and the same approach, the machine learning of this code, is helping us to understand how we work, how your body works, how your body ages, how disease generates in your body, how your cancer grows and develops, how drugs work, and if they work on your body. This is a huge challenge. And it's a challenge that we share with thousands of other researchers around the world. It's called personalized medicine. It's the ability to move from a statistical approach where you're adopting a Gaussian to a personalized approach, where we read all these books and we get an understanding of exactly how you are. But it's a particularly complicated challenge because of all these books, as today, we just know probably 2%. 4 books of more than 175. And this is not the topic of my talk, because we will learn more. There are the best minds in the world on this topic. The prediction will get better, the model will get more precise. And the more we will learn, every time we will be confronted with decisions that we never had to face before. About life, about death, about parenting. So, this conversation, and we are touching the very inner detail on how life works. And it's a revolution that cannot be confined in the domain of science or technology. This must be a global conversation. We must start to think the future we're building as a humanity. We need to interact with creatives, with artists, with philosophers, with politicians. Everyone is involved because it's the future of our species. Without fear, but with the understanding that the decisions that we will take in the next year will change the course of history forever. Thank you.
So when we finish the first draft human genome, all the results is really confused. So what is the meaning of those letters? What is the meaning of those A, G, T, C? So it comes with the questions, what if, because mutation on this position and that position, what is the consequences it might bring? So we start to look at a patient or an abnormal people. For example, we scan in the people who suffer from leukemia or lymphoma, several types of blood cancer, and compare their genomes with the normal people to see what is the main differences that the patient will share in common compared to the normal people. When we have the differences, we start to cause a mutation, not on human, of course, because it in mice, because it in cow, because it in monkey, to see what is the consequences of the mutation. What if we delete some nucleotide here, replace some nucleotide there, and see the consequences? And step by step, we now know pretty much about the human genome, especially the coding genome, which just take around 1.5% of the human genomes. So now, from the human genomes, we know which gene is causing, which gene is corresponding to the eye color, which is corresponding to the skin color, which is responsible for the shape of the skull, the height, the BMI, and you will see in this clip that scientists now can predict a human face just from the DNA genomes. So the application of this one is pretty interesting, right? Right now, a mother can know how her child look like when he or she come adult age, even when the child has not born yet. And now police can, based on the DNA sample from the crime scene, can predict the face of the criminal without any camera, any evidence, any witness evidence to see how this looks like, just from DNA. So let's come back to the basic fundamental background of genetics. What is DNA? So DNA stands for deoxyribonucleic acids. So it's made mostly from three main parts. It's a sugar, a phosphate, and a base nitrate. So a nucleotide. So DNA is a polymer of the nucleotide, and the nucleotide is composed three main parts. First is a 5-carbon sugar. Here, 1, 2, 3, 4, and 5. A phosphate group, which is attached to the carbon number 5. And we have a base nitrate. Which is attached to carbon 4. And we have 4 types in DNA. We have 4 types base digit corresponding to 4 types nucleotide, which is written in any genetic code of any organism. It is A for adenine, C, cytosine, T is for thymine, and G for 1. So in RNA, DNA is stands for deoxyribonucleic acid. RNA, the cosm of DNA, is stand for ribose. We just lost the nucleic acid. And the main difference between DNA and RNA is in here, in carbon number 2. So in here we have a hydrogen group. In case of RNA is ribose nucleic acid. Instead of DNA is deoxyribose. Deoxy, that means you lost oxygen. You lost oxygen in the hydroxyl group of carbon number 2. That's the only difference. But these differences bring to physical characteristic of RNA is it really unstable compared to its current DNA. So this unstable, if later any of you have to working with RNA, you have to work in really clean in a clean environment, because RNA is really unstable and easy to decompose in a normal environment. So, nucleotide is a monomer DNA, and when we combine its DNA, so we will have a covalent link between a two hydrogen group of the carbon number 3 and the phosphate group of the next nucleotide. So now we have another nucleotide here, and we have a covalent link between the carbon number 3, this group, linked with the phosphate of the next nucleotide. We still have in here H2, so carbon number 5, oxygen, I think, alpha oxygen, the other base matrix. Everything are the same. 3, 4, and 5. So this covalent link, we see that if we have looked from the top to bottom, we can see that in this strand of DNA, the 5 carbon come first, and the 3 carbon come later. And that's why we call this is a 5 prime, and this is the 3 prime of the DNA strand. Okay? Because in here, the 5 carbon come first, and the 3 carbon come later. Right? Here, we see that here is the 5 prime, and here is the 3 prime direction of the DNA strand. So among the 4 bases of DNA, in RNA we have also the 4 bases A, U, G, and C. And we have in RNA the T of DNA would replace by the U, stand for uracil in RNA. So the structure of U is quite similar to T, thymine. The only differences is in the carbon number 1. Instead of the hydrogen atom, we have a methyl group attached to the carbon number 1 of thymine, but in uracil we don't have the methyl group in here. That's the only difference. Okay. So, in 1952, Franklin, using X-ray technology to demonstrate the structure of DNA, we have also shot off with an experiment to quantify the type of nucleotide in the double-strand DNA. And he find out that also the shot-off rule that in a double-strand DNA, the number of adenines is always equal to the number of thymine, because A and T are always linked together by 2 hydrogen bonds here, and the number of G and are always equal to the number of C, while G and C are always linked together by 3 hydrogen bonds. Okay, so it leads to the concept that, for example, here we have a DNA, and we have a positive strand and a negative strand. So, what is a positive strand? What is a negative strand? So, please denote that in DNA replication and transcription, these two processes we will go for more detail in chapter 2 and 3. But in DNA replication and transcription, the newly DNA strand or the RNA always synthesized from the 5' to the 3', not the way around. Okay? It only been synthesized in one way from 5' to 3'. So, as a consequences, if we have 2 strands of DNA like this, the transcription factor, which is synthesized the mRNA, will come in and binding in which strand? The top or the bottom? The top. So the transcription factor will bind in this strand or in this strand. Which strand is being binding to start synthesize the mRNA?
First.
There.
5 to 3.
Yeah, from 5 to 3, but please take in this picture and tell me which strand is combining it. No one have an answer? Top or bottom? We learned this in high school. So we have a double-strand DNA. So the top is the 5' to 3', and the bottom is 3' to 5' in this example. Now I have a transcription factor here. Binding and start to synthesize the mRNA. It will binding in this strand or in this strand. The top or the bottom strand. that the champion factor only synthesized in the 5' to 3' direction. Yeah, so which strand is binding in here? Okay. So because if it only synthesized from the 5' to 3', so if the gene is beginning in here, so it will widen in the 3'. Transcription factor will widen in here and start to synthesize the mRNA from 5' to 3', right? So that meaning here, it will widen into the bottom strand, and because it's binding into bottom strand, so the new mRNA will have the sequence which is complementary with the bottom strand, which is identical with the positive strand, the upper strand, the top strand. So that's why in here, this strand, we call it the negative strand, or sometimes we call it the template strand. Is negative strand or template strand. And bottom, top, because it has the sequence identical with the mRNA, we call positive strand. Or sometimes we call it the coding strand. Okay? Remember this because it's important with your exercise. And since we have the mRNA, we will have a part of the RNA have not translated yet, but it is 5' ETR. Okay? Some of you have involved in the bioinformatics practical course. You remember the exercise where it is. Remember the exercise related to the partial translated, right? Which part of the exon is partly and which part is completely translated. So here, for example, here is a 5' ETR. We have the mRNA, but the translated, that means the ribosome, came in here and start to translate from this position. So all the region before is a 5' ETR. Okay. So let's have a small exercise to make you less sleepy. Please identify for me in here. We are designing primer. Primer is a fragment of DNA that combined with a certain DNA template. Okay. Using for sequencing technology. The required primer is it has to have a complementary sequence with this one. Note that in this sequence is writing from the 2' to 5'. So what is the sequence of the primer that has a complementary sequence of this segment in the 5' to 3' direction? Everyone finished? Anyone can read for me the sequence of the primer? Anyone finish? Yeah. That's correct. So in here, just simply put the chart rule because the sequence of the template are already written in print-on typewriter. So we just apply the chart rule. A is limited and G is limited. And the sequence the students have written for it. So now compare between the DNA structure and RNA structure. Yes, RNA is ribose nucleic acid. We have slightly differences in the carbon number 2, right? With the hydroxyl instead of the hydrogen atom in the DNA. Most of the RNA are single strand compared to double strand in DNA. And this is one reason why DNA is more stable with RNA, because it has a two complementary strand. And we have the uracil instead of thymine, uracil in RNA instead of thymine in DNA.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we've simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the bases attach to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair non-specific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
So in here I have made a small table to compare between the differences between DNA and RNA. So I don't know if in the slide I gave you in Moodle there is also this slide, right? No, okay. Sometimes I make a little bit of change. The slide I give you in Moodle and the slide I present you here, because I just want to know who is really going to lecture and who's not. But don't worry, it's the same. You can take a picture if you want. So this is one part that's quite important. So the student who not attend to the lecture class, I will not take the attendance, but they will have to compare by their own. Right? You want to take a picture? So let's go on the structure of genomes in different organisms. First is in the virus genome. Virus virus have a structure that is quite simple, that some scientists have not considered it as a living organism. It composed by only two things.
Hay là không?
Protein envelope, surrounding in DNA or RNA, acid nucleic in general, nuclear core. So the virus genome can be linear, or circular, or it can be single or double strand. It can be DNA and RNA, depending on the species. Some virus was composed by RNA genome, called retrovirus, because when it inject the RNA into the host cell, is merged using the host facility, the host mechanism, and the host ingredient to synthesize back from RNA to cDNA, and then from cDNA to transcript back to mRNA to synthesize its protein envelope to replicate themselves. That's why they call the retrovirus, because they have to reverse from mRNA to cDNA. And because of the very simple structure like this, normally the size of a virus is relatively small, just around 2 kb, to the maximum virus is 2 megabase per. That means around 2 million base pair. Just to compare, the human genome is around 3 gig. The two most famous retrovirus is the HIV and coronavirus. Right? These both have RNA nuclear core. And inside here, in HIV, it has also a reverse transcriptase, which will transfer the mRNA to cDNA in the host cell while it's infected into the host cell. So, for example, here we have a life cycle of an HIV virus. The HIV virus always attacks into the most important cell in the immune system, which is the CD4 cell. The CD4 cell is considered as a leader in the immune system. And it's infects the CD4 cell based on a special receptor in the membrane of the CD4 cell, which is GP120. CCR5 receptor. So if it connects to this CCR5 receptor, it's realized that this is the CD4 cell, this is the cell I have to infect into. It starts to inject all of this mRNA nuclear inside the host cell. With the host cell facility, it's using the reverse transcriptase to start to reverse the mRNA into the cDNA of the virus. So this is the copy of the RNA versions of the genome of this HIV virus. And it can merge into the genome of the CD4 cell and start to synthesize millions of copies of HIV using this cDNA copy. Synthesize the mRNA versions of the genome and also the protein envelope to wrap around the RNA genomes and replicate successfully millions of HIV and continue to express out and infect into the other CD4 cells. Okay. Now come to genome of another organism, which is prokaryotic cell. So the prokaryotic cell, a little bit different with eukaryotic cell, which is most of the genome of eukaryotic cell is single molecule. It have it also have each chromosome. Also, it have protein, not the histone like in the eukaryotic cell, but the other protein to keep protecting the DNA nucleic core. In a prokaryotic cell, we have also observed DNA outside of the nuclear core, nuclear regions we can call the plasmid. It's a very small fraction, normally just a few kb. So mostly, this plasmid was the gene encoding for the antibiotic resistance. So this figure is demonstrates the structure of the nuclear in the nuclear region in the prokaryotic cell. So we can see here the nuclear regions which does not have a membrane to separate the nuclear out of the cytoplasm like in eukaryotic cell. But the nuclei here were composed by the DNA combined with a complex protein. And this complex protein sometimes make the DNA in several different shapes. For example, in the shape of supercoiling, when the DNA is supertwist, it's more convenient with this shape when the cell is about to separate during, for example, during mitosis. And this is the scan of the microscope of the DNA, prokaryotic DNA, in the two different forms. In the relaxed one, you can see here a circular double-strand DNA shape, and in the supercoil, when the DNA was fused together in a different shape. And we can categorize the different shape of the DNA when we run into electrophoresis. Normally, the supercoil shape, as you can see here, the supercoil link, when it is fixed like this, is decreased its size, and thus it can move fastly in electrophoresis. So the supercoil will have the size less than in the shape of the nickel and the lighter shape.
Hình như cứ điền được mấy cái này là con nói này, cứ điền được mấy cái này làm được cái này là cả
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase 1, twists are introduced in the DNA molecule that causes it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleoli of the bacterial cell.
So compared to the eukaryotic genomes. So in eukaryotic like in the animal cell or in plant cell, we have the nuclear, was the DNA chromosomes was organized in a specific region called a nuclear region. And this region was separated with the cytoplasm by the nuclear membrane, right? Like the animal cell here and in the plant cell here. So this membrane will categorize, make the differences in the transcription process and also translation process of the prokaryotic cell and the eukaryotic cell. So because of the nucleus membrane, so if the DNA was replicate, it's also replicate inside the membrane, but if the transcription happen, the mRNA will synthesize inside, but later it will have to go outside of the nuclear membrane to start the translation synthesize protein. Meanwhile, in the prokaryotic cell, this process, transcription and translation, can happen the same. But we calculate later in chapter 2 and chapter 3. So, most of the genetic genome was, the DNA was structured in a linear double strand. And this linear double strand is combined with the histone protein to form chromosomes. So, each package of combination DNA and histone is called chromosome. Like in human, we have 20 pair chromosome, 23 pair chromosome. That means we have 46 chromosomes. That means 46 of linear DNA molecule associated with the histone protein. So,
Ra ngoài cạnh nhau.
we have DNA wrapping around a histone protein. And this histone protein, we thought is one histone protein, but in fact, just wrap around one around inside the nucleosome. So nucleosome in here is the structure of histone protein, which is octan. With octan with 8 subunits. So we have 2A2A subunit, 2A2B subunit, 2A3, and 2A4. So in total we have 8 subunits to compose 1 unit of the octan. And we have another histone subunit was binding in here, in different here, we have the H1. And H1 is called a linker protein. And if we wrap around another so H1 is linker.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are divided. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So the histone protein was play a critical role in epigenetic process, which will decide which gene was turned on and turned on during the development through a process called histone modification. But we will get into histone modification later on chapter 7. Now, for the moment, just remind that the histone protein is designed to compact with the DNA and twist one at a time to for example, when the cell is start going to cell division. So it needs a very compact shape, twist very neat shape, too easy to separate into different cells in cell division. And for example, in the other circumstances, for example, when the DNA want to divide, or there's a signal about transcript express the gene out through protein, it will have to uncondense, untwist to a relaxed form. So the DNA replication complex or the transcription factor complex can come into and have a space widening into the DNA to start to replicate or transcription. So, you see in here the DNA combined with histone, and in the dense shape, remember in the dense shape, it can combine and forms a chromosome shape as we see in microscope. But without the very dense and twist shape, it's really hard or difficult to observe the chromosome. So this is the most dense shape of the chromosome. In a most relaxing shape, it will have a shape like this. And this shape mostly is using in the DNA replication or RNA transcription. heterochromatin or acrochromatin. So in the heterochromatin is like in this shape. It is highly densely stained, which is most is happen in this shape where the sample is divided, and in the other shape chromatin when it's in this shape when the cell have a signal to synthesize the DNA or transcription mRNA. Now come to a new theory. The new theory is still have many arguments, but still have a widely agreed in the community of science. That is Angle-C White. So this theory came from a fact. That most of the eukaryotic cells, there is organelles like mitochondria and chloroplast, which have its own DNA. And when scientists compare this DNA with the DNA of the bacteria, they see that it's more or less the same. So, what kind of conclusion we can have? What kind of hypothesis? Let's say that we have not had any conclusion yet because we not based on experiment. But the phenomenon is in the eukaryotic cell, with the organelle like mitochondria or chloroplast in the case of plant cell. The DNA genomes of the mitochondria and chloroplast is similar to the eukaryotic cell. So what is the hypothesis here? So now you have observed the data. You observe an event and you have to make a hypothesis to the event. So what is the hypothesis here? I will not ask you to prove it because right now science cannot prove it also. But what is the most likely hypothesis you can pose in here with this observation? You understand what is the question, right? Alright. So, in the eukaryotic cell, we have the organelle like mitochondria. Mitochondria is, entity is synthesized energy for the cell, right? But it has its own DNA. It's different with the other organelle is it has its own DNA. When science sequence this DNA, they see that it's quite independent with the genome of the host cell, but similar to the genome of the prokaryotic cell. Okay? You understand the circumstances? So, what is the hypothesis in here? Yeah, please.
So I think, in mitochondria and chloroplast, they have the prokaryotic DNA. So I think that they might be an individual prokaryotic, and then they kind of like sync with the host body, and then they like through time they become one.
That is exactly the Angle-C biotic theory. So, the result scan of the mitochondria with the prokaryotic cell identify a largely identical level showing that maybe those organelle is prokaryotic original. And in some point of the evolution, these two organisms are combined together, which is called endosymbiotic. And the prokaryotic cells start to synthesize the energy for the eukaryotic cell, and the eukaryotic cell in the reverse combine all the other ingredients like nutrients and water and oxygen and all other ingredient stuff to the eukaryotic cell. Okay.
Endosymbiotic theory explains how scientists believe eukaryotic cells developed. First, the nucleolus was surrounded by the enfolding membrane, creating the nucleus. Then further enfolding membrane formed the endoplasmic reticulum. Then the cell engulfed an aerobic bacteria, which would ultimately become the mitochondria. Afterward, a cell with mitochondria engulfed a photosynthetic bacteria. This photosynthetic bacteria would become the organelle known as a chloroplast. Mitochondria and chloroplasts contain their own small supply of DNA, which may be remnants of the genome they had when they were independent bacteria. It is important to note that we know that aerobic bacteria were engulfed first because there are animal cells which lack chloroplasts, while both plant and animal cells contain mitochondria.
So in human mitochondrial genomes, so people start to curious about human mitochondrial genomes for a long time because it seems to be a very interesting tool to study about lineage, evolution, and migration. So mitochondrial genomes have some unique features compared to the nuclear genomes. Firstly, is mostly maternal inherited. That means that almost all the mitochondria we inherited from our mother. Although there is evidence that there's evidence that we inherited some mitochondria from our father too, but mostly from our mother. And somehow the genome in the mitochondria in humans is related to intelligence. When we annotate it, for example, if there's a mutation in the mitochondrial genome, it will cause intellectual disability. So there's another evidence to show that most of our intelligence we inherited from our mother than our father. Okay. So this is another piece of advice. If you want your kid, you want your children to get smart, you better get yourself a smart wife. Okay. Anyway, when we scan the human mitochondrial genome, we see that it composed by 16,569 base pairs. And it contains 37 genes, which is mostly encoding for tRNA. 22 of this gene encoding for tRNA. And only 13 of these genes remain coding for polypeptide. So the sequence was composed, have been fully published in the link below. So if you put this link in your web browser, it will link you directly to the revised reference sequence of the human mitochondrial genome.
Sau này chạy như chó có chút điểm này đấy.
So this is the map.
Thấy nhanh
You
như
see,
có điểm.
this is the map of the older human mitochondrial genomes. We can see here it has all the characteristics of the prokaryotic genome. For example, most of the genome was encoding for something. So it different with the eukaryotic cell because in the eukaryotic genomes, most of the genome is not coding for protein. Why, please? Like in human, we have only 1.5 or 2% of the human genome is encoding for something. Different with here, nearly 90%, more than 90% of the genome is encoding for something. Protein, tRNA, RNA, ribosome RNA. And we have only 2 regions that is not coding for RNA or protein. Is DLUC, or stand for the hyperviration regions. So these regions is using to identify the identity of an individual through from one generation to the next. Because this is the most vulnerable region compared to the other one, because if you have a mutation in the other region, normally this mutation will affect severely into the vitality of the carrier. So with the natural selection, we'll remove this mutation outside this region, leading to this one is the most hypervariant. So this one was applied because normally the mitochondrial genome is much more stable than the nuclear genomes. So these regions were applied to identify a material or a shoulder who was sacrificed a long time ago without any coffin or any proper body, so their DNA was seriously damaged. Why, please?
Thực ra về cái công nghệ mà giải trình tự cái hệ gen ti thể người này thì đầu tiên khi phát triển ở nước ngoài thì người ta ứng dụng cái công nghệ này để mà xác định cái phả hệ của những pharaoh mà đã qua đời cách đây khoảng 5-6 nghìn năm. Bởi vì khi những cái mẫu vật đấy, bởi vì là người ta đã qua đời còn cách đây quá lâu cho nên hầu như là cái bộ genome của công nhân là không bị hết. Tuy nhiên cái bộ gen ti thể này thường rất là bền vì nó là mạch vòng, nó khác với hệ genome người ở trong nhân nó là mạch thẳng. Thì vì genome ti thể nó là mạch vòng cho nên cực kỳ bền, cho nên người ta có thể ứng dụng cái đoạn giải trình tự cái đoạn DLUC, cái đoạn hypervariant này để xác định xem là cái phả hệ của ông bà này nó như thế nào, ai là con của ai. Thì khi về Việt Nam thì trong viện Hàn Lâm này cũng có viện công nghệ sinh học, tôi nhớ không nhầm là hình như là phòng của giáo sư Vika cũng ứng dụng công nghệ này để mà giải trình tự cái hệ gen liệt sĩ. Vì liệt sĩ cách đây qua đời thì cách đây sớm hơn rất nhiều, đúng không? Không đến 5-6 ngàn năm, cách đây khoảng độ 50 năm gì đấy. Tuy nhiên khi qua đời thông thường là do điều kiện chiến tranh nó chôn cất vội vã, không có quan tài, không có một cái hình thức chôn cất bình thường, cho nên là mưa nắng đã làm cho cái hệ DNA trong nhân hầu như là bị phân hủy hết. Cho nên dựa vào cái DNA nhân để mà xác định xem là hài cốt này là con của ai, xác định nhân thân là hầu như là không xác định được. Cho nên người ta phải dựa vào cái công nghệ DNA tinh thể này, vì là công nghệ DNA tinh thể tương đối bền. Dựa vào cái đoạn hypervalue này để xác định xem là hài cốt này là liệt sĩ này là con của ai, dựa vào cái người phả hệ ở bên mẹ, bởi vì là như tôi đã nói thì hầu hết tinh thể là di truyền từ mẹ. Okay, so now
Đấy trở lại, năm giám sản con này làm không phải. Ăn mắt, ăn nước. Có các cái dầu không? Có cái lô thì nó dầu.
Mitochondria, found in all cells of the eye, release energy from and stored in ATP molecules. Mitochondria carry its own DNA. Mitochondria were once free-living bacteria that were engulfed by ancestor of eukaryotes, but not digested. Bacteria and larger cell established a symbiotic relationship, in which the larger cell provided protection, while bacteria produced food. The mitochondrial chromosome preserves many features like bacteria. First, they are circular molecule. Human mitochondrial chromosome is only 16,549 base pairs long, which is not much larger than any bacterial plasmid. Second, the mitochondrial chromosome is tightly packed with genes. Unlike nuclear chromosomes, which have large intergenic regions of non-coding DNA between genes. Third, most mitochondrial genes lack introns, which are non-coding information within nuclear genes. Human mitochondrial DNA contains 37 genes. 13 genes are involved in process of oxidative phosphorylation. 22 genes encodes transfer RNAs for specific amino acids, whereas 2 genes encodes 2 subunit RNAs and ribosomes. Control region. The control region contains the signals to control RNA and DNA synthesis in mitochondria. It is also our fastest evolving DNA sequence. This hypervariable region accumulates mutations at approximately 10 times the rate of nuclear DNA. This region is also called D-loop, relating to structure formed when mitochondrial DNA replicates. Complex 1 genes. These 7 genes encode subunits of NADH dehydrogenase, which is an electron transporter, involved in production and storage of energy. Though the genes lie in mitochondrial DNA, the process of producing energy requires molecules that are produced outside of mitochondria. Complex 3 genes. This mitochondrial gene encodes cytochrome B protein, which is part of inner membrane of mitochondria, known as ubiquinol, or cytochrome C oxidoreductase. Complex 4 genes. These 3 genes encode subunits, which are part of cytochrome C oxidase complex. This complex acts as electron transporter during process of producing and processing energy in mitochondria. Complex 5 genes. These 2 genes encodes proteins of ATP synthase complex. This complex is located in inner membrane of mitochondria. Ribosome RNA genes. These 2 genes known as 16S and 12S are RNA, which are used to build ribosomes. During protein synthesis, ribosomes assemble to translate messenger RNA. Transfer RNA genes. These 22 genes encodes transfer RNA molecules. Each transfer RNA is associated with a specific amino acid. Transfer RNA plays an important role in protein synthesis, delivering their associated amino acid to ribosome for incorporation into new protein. The human mitochondrial chromosome has been vastly reduced. Over time, genes for functions that could be provided by the host were lost. Also, some genes needed for respiration were transferred to nucleus. Millions of years of evolution have resulted in small mitochondrial chromosomes. When egg and sperm unite, the sperm's mitochondria are discarded. Mitochondrial DNA is therefore inherited only from mother. Within a family, females from grandmother to daughter all share identical mitochondrial DNA. Unlike DNA in nucleus, mitochondrial DNA does not recombine with other chromosomes.
So in here I have made a small comparisons between the human nuclear genome and the mitochondrial genome. We see that in the side there's a big difference between nuclear genome, like in the slide in the clip you see. During evolution, the mitochondrial genome had eliminated several reasons and decreased the size of the genome. So up to now, we have the mitochondrial genome is just more or less 16,000 base pair. And the size of the human genome is around 3.3 billion base pair. About the structure, the nuclear genome is the DNA was combined with histone to form chromosomes. So we have 23 pair corresponding to 46 chromosomes in a nucleus cell. Meanwhile, in one cell we have thousands of mitochondrial genomes corresponding to thousands of mitochondria. The number of genes, of course, the number of genes in a human nuclear genome is estimated around 24,000 genes. In mitochondria, just 37. The size of the nuclear genome in human is quite large because it's a cell. It composes also the intron regions, right? So the size of the normally average size of the human nuclear genome is around 40 kilobytes, which is why one gene of the mitochondrial genome is just around more than 400 base pairs. The percentage of coding DNA in nuclear genome is just about 3%, and mitochondria is just 3%. And also the other features you can compose by yourself. All right. So what we need to remember today. So DNA is basic unity. DNA is stand for deoxyribonucleic acid, and it composed by four main bases A, T, G, and C. Complementary linked together, followed by the Chagas rule: A linked to T and G linked to C. So the DNA sequence and also RNA transcription is always happen from the 5' to 3'. So in virus, if the DNA, the genome can compose by RNA or DNA, but in prokaryotic cell and in eukaryotic cell, it all composed by DNA, double-strand DNA. And in eukaryotic cell, DNA is associated with a special protein called histone. One histone composed by A subunit, 2A2A, 2A2B, 2A3, and 2A4. Okay, so I think that is enough for today. Do you have any questions? Okay, thank you very much, and see you next week.
sách sẽ đến đây tuyển thành viên. Các bạn ở lại
tỉnh của thầy ngủ cho